endogenous in-tremble oscillation of 24 hours. And the circadian rhythm consists of three parts: the input part, oscillator, and the output part. So our body can sense the signals from outside and then generate in the oscillator. Oscillator is a self-sustained endogenous pacemaker, and it can translate these signals to oscillation into physiological behaviors. This is the output part. So actually, we have a clock in our body, and it tell you, tell your body what is the condition, and then tell you what you need to do. For example, we have highest body temperature at 7 p.m. Here you can see, and the lowest temperature around 4:30, and we have best condition for exercise around 5 p.m. So what is the molecular level? Why does this circadian rhythm can keep going? From the molecular level, we have positive regulators and negative regulators, and these proteins, they form a negative response loop, and the BAMO and the clock, they are positive regulators, and they form the heterodimer and bind to E-box in the promoter part of Per or cry genes or any other clock regulated genes. And after transcription and translated in the cytoplasm, they in return transferred into nucleus and inhibit the activity of clock and BAMO, inhibited their own expression. But after phosphorylation, these two proteins, they go to degradation. So the inhibition is released. And this way, they keep the clock going on and on. And in our labs, we are interested in the Per2, this protein. And this protein has a lot of functions. I just list several of these, only three here. Of course, this is a circadian rhythm protein, and have function in circadian rhythm. And also, it may have function in cardiovascular system, and also it's related to cancer, which we are really interested in this part. But we don't know the role how Per2 have function or worked as a tumor suppressor. So we have two oscillation systems in each cell. We have the circadian rhythm, we have a clock, and also the cell cycle oscillation. And several papers they already point out some circadian rhythm genes, they also have function in the cell cycle control. And our Per2 actually also worked as a bridge to connect the circadian rhythm and the cell cycle control. This data I show the new finding in our lab. We found actually Per2 have the function in PP3 pathway. You can see from here, Per2's function is to inhibit the ubiquitination of PP3. You can see the last two lanes. MDM2 is a well-known E3 ligase for PP3. If you add the Per2, it inhibits the ubiquitination of PP3 by MDM2. And also, these three proteins, they can form a complex. So MDM2, PP3, and Per2, they bind together. And we all know after cell cycle checkpoint activation, PP3 and MDM2 both phosphorylated and they separated, but we don't know how Per2 released from this complex. This is my job to figure out how it is released. So we already introduced a new partner for Per2 here, is MDM2. This is a well-known E3 ligase, and for the ubiquitination system, there are three important enzymes: ubiquitin activator, conjugate enzyme, and conjugating enzyme, and also the E3 ligase. These three enzymes are very important to add the ubiquitin to substrate, and after polyubiquitination, the substrate goes to degradation. So if MDM2 is the E3 ligase for Per2, some features I listed here. If this is a real E3 ligase, it can recognize the substrate, and at least it will bind to the substrate, and of course introduce or add the ubiquitin to the substrate. So the MDM2 did do all of this job for Per2? We didn't know. So first I checked in vitro whether MDM2 and Per2 they can bind together. For this experiment, we use the in vitro transcription and translation system to express all of these proteins, and we incubate them together at 37 degrees. And we use anti-flood beads to pull down MDM2 to see whether Per2 bind to MDM2. And we also change the ratio from 1 to 2.5 to 1 to 25. So we can see Per2 here, Per2 bind to MDM2 from the IP result, and with the ratio increase, more Per2 bind to MDM2, right? So this data at least show Per2 can bind to MDM2 in vitro. But we don't know which part of Per2 bind to MDM2, so we chop the Per2 into several fragments and make the fusion protein. All of these fragments we fuse the GST tag and did the GST pull down assay. And this layer is the input, the radio labeled MDM2. And from here we can see at least fragment 3 and 5, they can bind to MDM2, right? So we want to see they can bind together in vivo. So we did the in vivo experiment. We use HCD116 cell line, and we co-transfect the Per2 and MDM2 plasmid, co-transfect to the cells and check the binding. This part is input part, all of these plasmid they express, and this is endogenous PP3, and we get different cell lysate fragments. The T represents the whole cell lysate, and C is the cell lysate from cytoplasm, and N represents the cell lysate from nucleus. We also want to see where they can bind together. And the reason why we use MG132, because if MDM2 is a real E3 ligase for Per2, we want to inhibit Per2 degradation and to see the binding. And we can compare the result with or without MG132. So from the data here, we can see they bind together in the whole cell lysate, and at least they bind in the nucleus. The Per2 protein can co-precipitate with MDM2, and the PP3 work just like positive control. And then I want to know whether MDM2 added ubiquitin to Per2, right? So I started the in vitro experiment and use the TNT system again, in vitro transcription and translation system again. This is my input part, all those proteins expressed. The MDM2, flag Per2, flag PP3, and the PP3. And that panel, the right panel, I use in vitro ubiquitination system to detect whether MDM2 ubiquitinate Per2. So I incubate those proteins together and added the ubiquitin acetylase. And for the first lane, MDM2 and PP3, this is positive control for our experiment. And we can see the ubiquitination signal of PP3. And the second lane is a negative control. And we also want to know whether the ubiquitination is dependent on PP3. So we have two lanes with or without PP3. And we can see where MDM2 and Per2 together, we can see the ubiquitination signal of Per2. So it seems MDM2 can ubiquitinate Per2 in vitro. The lens in the green frame, I checked the binding between MDM2, Per2, OPV3. We use the same sample just as the ubiquitination set. And here we can see for the PV3 signal, because of the IgG, so the overlap between PV3 signal and the IgG. But we can see this is MDM2 signal. So MDM2 binding with PV3 together, and this negative control, and this Per2 and make MDM2 together, and Per2 make MDM. This is make PV3. Here we use make PV3, and this is make Per2. So they bind together. Now my job is to figure out whether MDM2 ubiquitinate Per2 in vivo. So I use cell line again. I co-transfect those plasmid into HCT116 cells. And this part, all the controls, Per2 alone, MDM2 alone, and PV3 alone. And this one, only this one is my real experimental one, and this lane is positive control, but the conditions are very critical. So till now, I do not have very beautiful figures to show you whether MDM2 ubiquitinate Per2 in cells, but I think it's promising if we compare the Per2 alone here and the Per2 and MDM2. These two lanes together, we can see around this part. This is the size of Per2, the ubiquitination signal. Seems like ubiquitination signal, but I needed to work more to figure out the best condition for MDM2 and Per2. And small conclusion, we found MDM2 and Per2, they can bind both in vitro and in vivo. And at least in vitro, we can see MDM2 ubiquitinate Per2, but in vivo, we need to work more. And I want to thank my mentor, Dr. Finkelstein, and all the other graduate students and undergraduate students in our lab. Thank you.
I have a question. This MDM2 protein is very well known as playing a role in cancer. How about those periodic proteins? Are they in cancer as well?
Pardon?
Those Per2 and other proteins, are they play a role in cancer as well?
Oh, yeah, the new finding shows Per2 worked as a tumor suppressor. So, yeah, I believe Per2 worked in cancer. Per2 have a real important role in cancer suppression.
Anything else?